Folks, it is my honor today. It is my privilege to welcome on the incredible Dr. Oded Rechavi, who is rewriting the scientific rules of the entire world. Oded, I think that's a fair way of saying it. From Tel Aviv University, award-winning neurogeneticist, redefining everything we know about heredity, memory, even ancient history. He's a scientist who's proven that experiences like starvation and immunity can be inherited not just through DNA, but through other mechanisms, turning what we considered as inheritance of traits that were thought to be nonsense for a smidge, turns out to be real. He uses sequencing to piece together a piece of the Dead Sea Scrolls. So there's a historical perspective. There's been turning of drug delivery mechanisms, publication upon publication. He's also created the most engaging science conference in the world called Woodstock Bio. First major conference to have a walk-on song, to have on one slide and to be non-published data, advocate for science communication, engaging on his social medias, recipient of the Blavatnik Award for Young Scientist, prestigious Schmidt Science Award for the Polymath Award, and now he's with us here on Twitch. Oded, welcome.
Well, after this introduction, I have to quit. I can only go down from here, but thanks a lot.
Thank you so very much for joining us today. Would you mind giving us just a little bit of background on how you got into the sciences in such an extremely diverse and such a cool set of like model organisms?
Yeah, so how did I get to science? That's also a little bit of a story. So I didn't grow up thinking I'd be a scientist. I mean, many scientists always say that they knew from since they were very young that they're going to be a scientist. That's not my case, in fact. That's not my story. I don't know exactly what I wanted to be when I was a kid, but when I was already a young man, I thought I'd be an artist, in fact. And I went to, I traveled to Paris, studied a little bit of art for half a year approximately. And then when I was sitting there in the coffee shops of Paris, it's actually a particular coffee shop, and I was thinking what to do with my life and how to continue. And I also looked at the brochure of Tel Aviv University of the different professions that different subjects you can study. And I saw that, and I thought of studying philosophy and psychology. But then they opened a new track for neuroscience, which was psychology, philosophy, but also biology, a few courses from different fields that are related to neuroscience, and I thought it sounded more cutting edge. So I started learning that, and in parallel also try to be an artist for a little bit. I had a couple of exhibitions in Tel Aviv, and then when I was studying these subjects, I realized that I love biology more. And then one thing led to the next. I was interested in neurobiology, so I wanted to, particularly because I wanted to study memory. But for a long while, until recently, actually, I didn't study proper memory, the way most of us think of memory as as the memory that is encoded in our brain. But in fact, I got sucked into studying other types of memory, in particular transgenerational memory, and that was kind of by accident. So I was studying in the first degree. I was studying neuroscience, and my conclusion was it was all very interesting, but that we don't understand the brain, and in particular, we don't understand memory. And so after I finished my PhD, did totally different things. I wanted to try to study this in a very simple animal, in the simplest type of brain. And this animal is the nematode C. elegans that I started studying in my postdoc in Columbia University with Professor Oliver Hobert. And when I got to his lab, my original intention was to study a particular type of memory that's called imprinting, but I ended up studying transgenerational memory. And it took me 15 years. Eventually, I also got to study real memories in the brain, and we can talk about that as well.
So when you're picking out that topic to study, how did it rabbit hole into transgenerational memory versus other types of memories? Because that also seems like you're saying, we don't understand the brain now, right? We didn't understand the brain then. So how does one come across such a thing to study? Like, how did you know to set up an experiment in a way that you could test for this kind of thing? And then at what point were you convinced that you had found something real? Because it also, right, like, in the TED Talks that we've watched that you've presented, it's kind of almost a little bit Lamarckian, right? And so it's like, how does one know that what you've found is real?
Yeah, so I'm going to organize it all, because at the beginning I just set up the background, and now we have to dive deep.
Yep.
Because you're right. First of all, I'll say that Lamarckian inheritance of acquired traits, inheritance of memory, I might argue is the most controversial topic in the history of biology.
Wow.
At least it's high up there in the sort of things that you can't say and that everyone assumed to be wrong. And there's a very long history for that. We'll get to that in a second. Okay? Let me tell you how I got to study. So I was finishing my PhD. I wanted to find the topic for my postdoc. I knew that memory is the topic that interests me the most, and we don't understand it in humans. So I said, okay, let's study it in a very simple organism, and that organism is C. elegans. The reason that the nematode C. elegans, this simple worm, 1 millimeter long, very simple, is such a good model for studying many topics in biology in general, but also neuroscience in particular, is that it has a very small and simple brain. So it has just 302 neurons always, so 959 cells in general, 302, third of the cells approximately are neurons. And we know, and they are largely, the nervous system is largely hardwired. So we know each neuron by its name and with which other neurons it is connected. We have a map of the entire connectome, like a subway map of the brain of the worm. And this is known and has been deciphered for more than 30 years. And the worm is transparent, so you can see the neurons fire using certain techniques. And it's also very convenient for doing genetics, extremely powerful for doing genetics and transgenerational memory, transgenerational studies, because the generation time between each generation from the moment the worm is born until you have the next generation born is just 3 days.
Wow.
That's very fast.
Yeah.
So in one PhD or one postdoc, you can do hundreds of generations. You study this in mice. First of all, their brains are unbelievably complex with billions of neurons wired differently, between every two mice, and also across life, the connections change. And also, it takes a while until you have the next generation. The C. elegans is really perfect for studying these two topics.
So, question from chat, Oded, just on that topic on its own. So, with these neurons, when these animals are learning, does that mean that there's no new connections that are formed between the neurons, that you're just kind of set, like, this is the nervous system, this is the connectome, you're not going to have changes in that?
Yeah, that's a very good question. So, we don't know enough about that to say, but largely when you think about, I'll be more accurate. Normally when you think about memory, and people assume that it works, is that when an animal learns, the connections between particular neurons are strengthened or weakened. The synapses, these connections between the neurons, become more, they fire more or less. Okay? In C. elegans, we have a few such cases, but not too many papers on that, in fact. Okay? There could be other simpler types of memories. This probably also happens, and there is evidence for that. But you could imagine even simpler mechanisms of learning and memory. For example, assume that the worm knows how to recognize a particular odor. He's born with the capacity to sense this odor. The worm senses thousands of odors. It's mostly, most of what it does is search for odors, that signal for food or enemies or dangerous things, toxins. Imagine that the worms knows how to recognize a particular odor, and you pair this odor with something bad, like starvation. And the worms can learn now to dislike the odor. And this has been shown. There are such studies. This can happen in theory by strengthening or weakening of synapses, like we just discussed. Or alternatively, maybe there's just less of the receptor that binds to the odorant molecule being made. And you can achieve the learning perhaps just by gene expression changes by having more or less receptors. That's an alternative mechanism of learning and memory that could, that might be happening here. And as I said, there are a few examples for synapses being strengthened after learning and memory in C. elegans, but not too many papers about, and if I go back to the question about totally new connections between neurons that don't know how to fire before, now suddenly they are connecting because of learning. I don't think that there are examples of that in C. elegans.
Okay, so it's, even though the way you outline it too, it's like it's simple, but it's still not quite simple, right? It's as the simplest that we can get currently with this kind of like small number of neurons set. You still have the complexity of learning, and I really like how you described it's not necessarily just neuronal firing too. It's you can change it just with the receptors. So it almost feels like Yeah, like that's learning, but that's not how we maybe classically think of as a learning. It's not necessarily embedded within the neuronal firing.
Right. So for example, exactly as you said, it's simple as it gets pretty much in neuroscience. 302 neurons, fully mapped connectome, and so on. But still, we know, for example, that there are many things going on off the grid. Neurons secrete all kinds of things, neuropeptides that act in a distance. So even though it's that simple, it's still complex, and we really didn't figure it out yet. That's also, makes you a little humble with the attempt to understand human brain or even the mice brain or even a fly's brain. It's just very, very difficult. And there are amazing achievements in neuroscience, especially recently when you see how even treating the brain as a black box, just recording with many electrodes can produce amazing things, like someone can move a robotic arm with their brain, but we don't understand how. All we know is the pattern of the activity that we recorded is correlated with the person's wish to move his hands, because he reports this hand. We train the machine to recognize this wish. And then when this person thinks, we don't know how this is encoded in the brain, but when this person thinks, move your head, move my hand to the left, we already trained the computer to recognize this pattern. We don't know what this pattern is, or how is it calculated, and so on, but we know to correlate it with the movement of the arm. So we can do amazing things treating the brain as a black box, but it doesn't mean that we understand it.
And it's really, like you said, it's humbling, because as long as we've known the exact number of cells in a nematode and the exact cell divisions and how they divide and what they do, and even the connectome, it's really humbling that we don't know how this works, like just how this nematode thinks.
Right. Exactly. You mentioned a few other things that I didn't mention. For example, we know from a single cell, the fertilized egg, the entire lineage, how it will, every cell, to what cell it will develop, and this is totally figured out for a long time. There have been a few Nobel Prizes awarded. So since the year 2008, Nobel Prizes to C. elegans researchers on different mechanisms discovered. So it's a very successful model organism. Many people, especially if they're not biologists, they hear, oh, it's a worm, you're studying a worm. No, this is a very, very powerful worm. Okay. And most of this is, if there's one message that I want, the audience to know, that is, discoveries in biology are made from simple models, from worms, flies, bacteria, viruses, fish. So the model organisms are very, very powerful for making discoveries. So what I was saying is, so the worm is so simple, yet complex nevertheless. And, also some of the things that I said, I cheated a little bit. For example, when I said that the development of the brain is totally hardwired, that the connectome is totally figured out, that we know each neuron with which other neurons it will form connections. This is almost true, but not entirely true, because there is variability between worms. People first created this connectome, this map of the connections of the brain of the worm, a long time ago using electron microscopy. And this was a lot of work. It was really difficult to do. And for a long time, no one wanted to do the second connectome, But when they did, they saw that there is some variability. So it's even more complicated than that.
And it's weird, right? Because when you're working on what is called the wild type worm, or the control that everyone bases against, in theory, they're all genetically identical, right? That you make your mutant in this one genetically identical worm, and then in theory, it should be linked back. But it's really cool that there is that variability to it.
Yeah, that's also a whole new thing, a whole another thing, that yes, we understand the worm C. elegans so well, but we know that even so the worm that everyone studies, it has been isolated in Bristol, England, many years ago. And this is the lab strain that everyone studied. We know that if you I'm not even talking about different types of worms, okay? It's still C. elegans, but isolates that were collected in different parts of the world, in many cases, are different in many ways. For example, oxygen sensation and collective behavior in C. elegans responds to. So if you elevate the oxygen levels, the worms, they group together and they lower the oxygen concentrations, because they just, they huddle together. And this is different in pretty much all other worms connected from other places in the world, other C. elegans. Okay, so this is a result of lab domestication. Many of the phenomena that we understand in C. elegans, including the ones that led to Nobel Prizes, like RNA interference, we'll talk about that, I assume, a lot, because this is the mechanism for transgenerational inheritance. That's totally different in other worms, isolated from Hawaii, for example. Hawaii is the Africa of C. elegans. This is the largest variability of worms. So their RNA in many cases works differently. In some cases, it's not inherited. In some cases, it is. So if Sidney Brenner, who started the work on C. elegans, started working on an isolate that he collected from Hawaii instead of, which is more wild than the one in England, more natural, then maybe we wouldn't discover RNA.
And so can these two worms, the one from Hawaii and the one from Bristol, can they interbreed? It's like,
yeah, absolutely.
So they're
it's the
same species, but the biological principles would be different that we learn.
Some things would be different. So, for example, this worm doesn't inherit RNA. The main worm from Hawaii that everyone studies doesn't have RNA inherited. We know a little bit about that. So that's also about when people say what can happen and what can't happen based on studies, it's very limited in many cases. Like if you think about what some organisms where people say that RNA cannot be inherited, transgenerational effects cannot happen, maybe just the wrong, organism. That's also a possibility.
Yeah.
I would also say that, a little bit of respect for the worm. I think we're building it up, but it's good that 4 out of 5 animals on this planet is a worm. If you just count the animals.
Wow. Okay? 4 out of 5 animals on planet Earth are a worm.
Yeah, I can put a link later to the Nature paper from a few years ago where this line exactly is written. That was a sort of a survey of animals in the soil or something like that. I don't remember exactly. And this is the quote from there. So if you just count the worms, not the diversity, not the types of worms that of animals that you have, just numeric. If you count the animals, 4 out of 5.
Wow.
So again, if you study worms, you're studying something very relevant. And we do it not because we're necessarily interested in worms, because worms have amazing biology. It's easy; they are easy to study, and also they affect everything. So the worms in the soil, they eat bacteria, they eat fungi, and just if you take away the worms, the world would crumble because they affect literally the gas exchange with the atmosphere because of their metabolic activity and so on. So this is important on every level.
And it's really wild then to think about that it was almost the right worm chosen out of all the possible isolates, that worm that was chosen that we've learned so much about biology from.
Right. So I don't know, it's not the only one. There are certainly others, but yes, they were lucky. We were lucky on many aspects that it was C. elegans from Bristol, England that was selected. Just pure luck. And there are many examples for these type of things.
And so when you picked because one thing that we usually get from the community is like, how do you pick the organism that you study, right? For your research question. It's also like the ideal model for you too, but like you could have picked any other organism too, right? To be able to work on. So it also like pushed you in the right direction as well.
Right, but by accident, because I said I want to study memory in the brain, and therefore I'll go to do my postdoc with Oliver because he studied C. elegans and they have such simple brains. But I ended up studying transgenerational inheritance, which wasn't my intention. That was also, yes, I it was a good idea to study C. elegans, but I was also lucky.
Yeah,
totally lucky. Okay, so I went to Oliver's lab and I wanted to study memory in the brain. And the reason I did this is that Oliver published a paper in 2005. I joined his lab in 2010. He published a paper in SciAnts about imprinting in C. elegans, which is a very particular type of long-term memory. Imprinting is a long-term memory. The term was, there's also genomic imprinting. It's a different thing. It's connected to epigenetics. We're not talking about that. Here we're talking about behavioral imprinting.
Is that similar to the concept of like baby goose?
Exactly. So the baby goose experiment, these are Konrad Lorenz's experiments. Who got the Nobel Prize and studied and coined the term. And what he has shown is that imprinting is a type of long-term memory that you can acquire, different animals can acquire in particular periods of their life. For example, if you're talking about geese, when they just hatch out of the egg, they start following the first moving stimuli that they see, and they get attached to it. And this attachment is formed a particular type of period in their life, and then they will just follow it, and this memory of who they are attached to will be very strong and will last for a long time. And it can typically it's their mother, but even if you put a robot next to them, they will follow it. And another type of imprinting is filial imprinting, the connection between the baby and the mother in general, or if you know about salmons swimming against the current to the place where they were born, that's another type of imprinting. So in that paper in 2005, Oliver showed that worms can also be imprinted. And typically, if worms learn something, C. elegans, you can teach them things. That's what attracted me to study C. elegans in the beginning. You can pair, like we said in the beginning, we can pair an odor that they like with something bad, then we learn to dislike the other. So they can learn even with a simple nervous system, but these type of memories typically are forgotten after just 2 hours. The worm lives in the lab something like 3 weeks, but they forget very quick. And that's a little disappointing, okay? But in that paper, Oliver showed, together with another researcher, Jean-Jacques Remy from Marseille in France, that if they learn certain memories at a very specific time point, one transition through 4 larval stages before they become adults. If you teach them the association at the first larval stage when they are very young and small, then it lasts longer. And in this paper, documented even memory of 24 hours. So I went to study this, and this is why I wanted to join Oliver's lab. I thought not only it's a memory, and in that paper they really they learned a lot about the mechanism. They say it happens in a particular neuron. One neuron out of the many is responsible here. It's a neuron called AIY, and there's this receptor there which is crucial for the memory. So wow, finally, something very molecular and impressive about memory. So I wanted to study that, but when I got to Oliver's lab, Jean-Jacques Remy, the collaborator of Oliver, worked in France, published another paper just when I started the lab, and he said that not only can the worms be imprinted, but that in certain situations, if you do it for a few times, then the next generations are also already born with the memory. And that was really shocking. It's a paper in Current Biology from 2010, and the paper is not mechanistic at all. It's just one figure, a scheme of the behavior. So I thought I want to study this, but I didn't want to study this behavior, which is complicated. We don't understand it too well. I wanted to study something much more molecular. So I thought, which type of, and I knew that, DNA can't do this. And in general, we know, and this is why it's so controversial, that we know that it shouldn't happen. Okay? Maybe it's important to say this again and stress this many times. There's a very long history to controversies about transgenerational inheritance, Lamarckian inheritance of acquired traits. There are different words for that. It's not all the same. It's not really even Lamarckian inheritance. Just for the non-biologists, I'll organize this a little bit before we talk about what we found and how we got to that. So Lamarck, a very important scientist, working in the end of the 18th century and the beginning of the 19th century, one of the first to use the word biology, one of the first to have an organized theory of evolution, which was based on the wrong mechanism. Lamarck published his papers on the subject. Darwin knew of them and published his own theory of evolution 50 years after Lamarck, and he was well acquainted with Lamarck's work, also referred to it, okay? But Darwin had the right mechanism of evolution, which is survival, which is natural selection. And Lamarck, what people remember from Lamarck, or people associate Lamarck with, is this idea of inheritance of acquired traits. In fact, that's not really accurate, okay? Everyone in Lamarck's time believed in inheritance of acquired traits. So he didn't even talk about it, really. Okay? He took it as obvious, as did many other people. Also, Darwin believed in the inheritance of acquired traits, at least for some of his life, and he wrote about it, including in certain versions of The Origin of the Species. And in many aspects, he was more Lamarckian than Lamarck. Okay. Lamarck thought that this is happening, but he didn't really talk about generations, about inheritance. So he focused more about mechanisms such as use and disuse. When, according to Lamarck, an animal uses a particular organ, it gets strengthened and kept. And when an animal doesn't do that, the organ goes away. Okay? And the famous example is the giraffe's neck. Okay? So there's always how Lamarck is explained. I'll say about it something. So according to Lamarck, the giraffe got its long neck because the treetops were high, and the giraffe used these internal forces to extend the neck towards the trees, and the next generations were already born with this long neck. And we know this doesn't happen, right? In contrast, the Darwinian explanation, natural selection, is that there were many giraffes, and they differed just by chance in the neck's length, and the ones that happened to be born with the long neck, you didn't know about genes or genomes or mutations or any of that. That was before. They just get to survive because they eat, the rest die, and the next generation, they share the same traits, so they also have long necks, and this trait takes over the population. About the giraffe, Lamarck only mentioned the giraffe's necks twice in his entire writing, and people picked up on that. Okay?
Yep.
even added it sort of a footnote, in a sort of later proof or something to his book. So it's not really something that he emphasized. Lamarck was discredited already in his life and also later ridiculed, and he's probably the most famous biologist or scientist famous for being wrong.
Okay.
But he had many contributions. You can also, argue whether that's the worst fate one can have, because many of his contemporaries, no one knows about them, but still we still talk about him. And there were also grains of truth in some things that he said. People objected him in his life and also later because he said a lot of nonsense, unrelated nonsense as well. For example, he rejected the chemistry that people already believed in his time and believed everything happened because earth, wind, fire, and water, although they already knew a lot more about that. And he also predicted the weather and made horrible predictions and did all kinds. He was a big theory type of guy who tried to explain everything in the world, from geology to biology, everything with big theories and ideas. And so he was wrong many times, but he also captured something like the march of life, the evolution. So he talked about that, and that is very important. And maybe he's the first to talk about really evolution, from simple organisms to complex organisms. Another something that is really Lamarckian is this emphasis of a march towards complexity. And but people remember inheritance of acquired traits, although, as I said, everyone believed it at this time. Okay, and now we're talking about, we have to distinguish inheritance of acquired traits from inheritance of parental responses or effects of parental responses on the next generation. Transgenerational inheritance versus intergenerational inheritance. We'll talk about that, okay? And also epigenetic versus genetic mechanism.
It almost seems like from the chat pointing out as well, that there is an association with the original term of acquiring of inherited traits, acquisition of this, that almost is loaded in a way where you have to kind of redefine the terminology and have a different term so that you don't have the biases associated, like we said with the long necks, because the transgenerational inheritance and intergenerational that we'll be talking about isn't about the long necks, but it is about maybe some kind of phenotype or something measurable that you can inherit. And so it's interesting about the historical context that we have the findings in.
Right. And there are a few, it's a great question, a great comment. And I mean, we can look at it from different directions. We can just say, let's forget about the history because it's so loaded, complex. And, immediately, if you say inheritance acquired trait, if you say transgenerational epigenetic inheritance, if you mention Lamarck's name, you get into trouble. On the other hand, you also learn a lot from the history because they discussed all of it. The discussions that we have today about what's possible or not, many of them were done in the past. And it also gives you great ideas. If you talk about, how do you find interesting topics? One of the principles is you intentionally take something that no one thinks can be possible and you examine it, and maybe there are some grains of truth. So, for example, one of the important reasons Lamarck was mocked, and has such a bad name is that he had a sort of history, complicated history with Cuvier. Cuvier is is a very important contemporary scientist of Lamarck. And he, the father of paleontology, and he wrote the deadliest eulogy in the history of science about Lamarck. Something like 8,000 words or something like that, of just destroying Lamarck systematically. Okay, he was afraid to publish it during his life, but it was published later. And there he some of the things that people say about Lamarck today are already traced to that eulogy.
Wow.
For example, many times when people want to mock Lamarckism or the idea of inheritance acquired traits, they talk about the role of the will of the animal to, the will of the animal as something that directs inheritance and evolution. And this is traced really to Cuvier. He picked on one example where Lamarck stumbled in his words to say something that sounds like that, and made a point of emphasis out of that. Okay? And people still today, in many cases, when you see also lots of the important figures in the history of biology, like Wallace and Darwin and others, when they talk about Lamarck, when they wrote about Lamarck, they talk about this as if that was his mistake. Okay? Cuvier, to make things even more complicated, he believed in inheritance acquired traits, just like Lamarck. The point where they differed is that he, unlike Lamarck, was a creationist. Okay? So Cuvier, a great scientist, many important contributions, much more systematic, unlike Lamarck, does proper science, but he's also but his real problem with Lamarck was evolution. Okay? So that is interesting. So it's important to know the history also when you talk about what is possible in inheritance of acquired traits. We'll talk about it in a second. What we found, what others found, and then you go back to the most controversial questions. Can what an animal wants direct its inheritance? You go back to Cuvier and you ask, this is maybe, this is the most controversial thing. Someone can say, let's study. Let's see if there's something there.
A willing into existence of a feature to survive better or to outcompete, right? Like I want this, so I'm gonna get it.
Yeah, or I'm thinking about it. This is what can thinking affect the heritable materials, for example. So the history is a very interesting source of ideas.
And it's interesting too, like you're in this field that you decided to do or fell into transgenerational inheritance, but there is also, right, other transgenerationalists like McConnell threatened by the Unabomber.
Sure.
And
right, I mean,
and you ended up in this field though. Like it seems right, almost like a landmine to go into. Like it almost seems like it's riddled with controversy because of just like the historical elements to it. So it almost seems like your science has to be on point when you're going in to study these questions and really controlled and rounded in a really, I mean, as science should be, but it's a much more intense method. It almost feels like.
Right. I think you can always be wrong. But so you might as well, try to tackle something difficult and, but I totally agree. I mean, McConnell, as you mentioned, a scientist who was really, attacked by the Unabomber, sent him a bomb, and there are other examples. I mean, Kammerer shot himself after he was accused of fraud, early 20th century. And the entire field of epigenetic inheritance has this huge dark cloud around it because of what happens in the Soviet Union, where under Stalin, Lysenko, totally forced communist ideology on biology, rejecting Mendelian inheritance and normal genetics, saying it's bourgeois. And, in fact, the inheritance of acquired traits and this idea of epigenetic inheritance fitted the agenda because everything can change into anything. You can take any man or any field and turn it into whatever you want. It will be inherited. It's not predestined and so on. So and that led to the starvation, the huge starvation, death, and also to many scientists were sent to exile to Siberia and killed. So it's extremely controversial, and this is why you have to handle it with care and be ready to face the criticism and do replicable work.
It's the historical context is really cool to be able to go over before we delve into the mechanistic of the science things. I think it sets the stage really nicely of just, again, the fact that you fell into it, like the chances of all this happening and falling into place, and now that based off your work, we're bucking what the original hypotheses are. It's just, we're lucky to have you, Oded, and we're lucky that you fell in this rabbit hole.
Thank you. It's totally my pleasure. And of course, I'm not the only one. There are many people studying this, and it's a big field. But okay, so back to maybe my story. So there was this paper I wanted to study how it might happen. So I was looking for a sort of a simple phenotype and a simple mechanism that could explain how this works. I knew that this type of inheritance of parental response across generations, let's not say inheritance of acquired traits for a second, like that the life history of the parents would affect the next generations even when they are unexposed to the environment that their parents faced. And maybe I'll define epigenetics here for a second, because people say epigenetics about many different things. For the sake of this conversation, the definition will be that it's inheritance. Epigenetic inheritance is inheritance across generations, because there's also inheritance across cell divisions. Let's put that aside for a second. Inheritance across generations that doesn't occur because of changes to the DNA sequence, right? But by other mechanisms, the star mechanism, they will be RNA, heritable RNA. And we should also distinguish transgenerational epigenetic inheritance from intergenerational epigenetic inheritance. Okay? Let's explain first intergenerational inheritance. One famous example of that is that during the Second World War, the Nazis starved large parts of the Netherlands, and the descendants of the women that were starved for half a year, they had the calorie restriction. They are different in some ways. For example, they have more tendency to develop diabetes. They have more schizophrenia. And this is because their mothers have been starved. This is not in the DNA sequence. However, for now, I mean, maybe in the future it will change when the research continues, because this is a huge epidemiological study. For now, we don't necessarily need to evoke a new mechanism to explain it because it is categorized under intergenerational inheritance. What does it mean? The comparison here was, what the scientists compared was the children that were in utero while their mothers were starved to their siblings, and also the next generations, so not just the children, but also the grandchildren. The reason we don't need to evoke a new epigenetic mechanism for that is that the children were themselves affected by the starvation. They were in utero being starved, so they are directly affected. Not only that, also the next generation, the grandchildren, are directly affected because the kids, while in the mother's womb, they already have germ cells. So their germ cells, sperm and egg, are also directly exposed to the starvation. So it's not in the DNA sequence. Okay? It is affected by the environment, but you don't need a new mechanism to explain it. In contrast, what we're studying are transgenerational effects, which are effects that continue for long durations, for multiple generations, also to generations that were not directly affected by the environmental cue, or stress, or whatever it is.
So the germ cells for the transgenerational one weren't all being affected, right? So it wasn't that the cells weren't exposed to any kind of stress, the organism wasn't exposed to stress, yet you're having some kind of inheritance across time.
Right, and that's again an advantage of SciAnts. You can study many generations. It's fast. You can do all kinds of genetic tricks to ensure that the germ cells are not affected directly, and so on. And then if you have these effects, these transgenerational effects that are long-lasting, also to descendants that were not affected by the environment, then you have to ask which type of molecule can do it. And this molecule, I mean, it's not DNA. We know that DNA sequence doesn't change in response to the environment in predictable ways. It changes randomly by mutations of different sorts, including big changes, but they are not directed and they're not predicted. And so whatever it is that's not DNA has to do two things. One of them is it needs to replicate somehow. Everyone knows, even not professional biologists, that the secret for why DNA is the heritable molecule is that every strand can serve as a template for the synthesis of the other strands. You can always perpetuate and synthesize more and more of it in every generation. It can contain information. You can pass on this information more and more. Doesn't dilute. So this other molecule that potentially transmits inheritance of parental responses needs to also be able to replicate. Okay? And or at least there should be some feed-forward process that perpetuates it.
Right, because you have to be able to maintain it. So presumably that takes out, like, let's say, a protein.
Right. The protein will dilute very fast. If you're talking about C. elegans, everyone produces hundreds of babies. Everyone produces approximately 250 or 300 worms. So after 4 generations, the dilution factor is 250 times 250 times, that's 4 billion, totally homeopathic, even after 4 generations. Nothing's there, okay? Certainly not after more generations. So you need to amplify. So that's one thing. The other thing, and there we are going again to the history, many of the interesting epigenetic responses we're talking about need to do the following. They need to move from somatic cells that interact with the environment and the change in response to the environment, to the germ cells. Okay? In our bodies, we have germ cells, and they are the cells that carry over the genetic information across generations. Sperm and egg. All the rest of our cells, brain cells, mast cells, all the other cells, what happens in these cells to the DNA, we're talking about DNA, doesn't matter to the next generation. We have the same genome in every one of our cells, equipped with, all the instructions for making all the proteins that we need. But if we have a change in the genome of, let's say, the mast cells, if I go to the gym, okay, and let's say that for some reason the working out in the gym changed my genes, it wouldn't change my genome, okay? It's not, okay? But let's say that for the game, if I went to the gym and it changed my muscle's genome, it doesn't. I have to, make this explicit. But even if it did, then it wouldn't matter to the next generations, because the next generations would only inherit the genome of the sperm and the egg. That's it. What happens in the soma stays in the soma. This principle is called the Weizmann barrier, after August Weizmann, again, a scientist, very important scientist from the Weizmann from the 19th century, and one of the people who resisted Lamarck the strongest. Okay, so if we're talking about, for example, some response to the environment that is carried on to the next generation, which is initiated in the soma, in somatic cells, we don't know of a mechanism for how it would directly change the DNA in the sperm and egg so that the next generation would be affected. In contrast, we can imagine another molecule which does transfer between different cells, including from the soma to the germline, and therefore would be able to carry somatic responses across generations. So that was a very long introduction, and now I'll explain what we found. So what we found is, so it was known in C. elegans that if you introduce into the worm different ways of doing that, double-strand RNA, not DNA, it would lead to silencing of mRNA molecules that complement this RNA that you introduce into the worm. You can introduce this double-strand RNA in many ways. You can just feed the worms on double-strand RNA and bacteria that produce double-strand RNA. You can inject them with double-strand RNA. You can soak them in double-strand RNA. They take this double-strand RNA, and this RNA, which has two strands. Normally, when we think about RNA, we think about just one strand, like the mRNA. DNA, double-strand RNA, double-strand, but with the double helix, everyone knows that, okay? Normally, RNA, when we think about it as a single strand, although there are also double-stranded RNAs. But if you introduce long, perfectly double-stranded RNA into the worm, it will be chopped into small RNA pieces, and these RNAs, small RNAs, will find now complementary messenger RNA molecules and will lead to their destruction in many different mechanisms. And this is a phenomenon that was called RNAi, or RNA interference, or gene silencing. Fire and Mello, two worm researchers, discovered it in a paper in 1998 and got the Nobel Prize in 2006. This year, an additional Nobel Prize was awarded to two other C. elegans researchers, Gary Ruvkon and Victor Ambrus, for finding natural small RNAs that also silence other genes that are called microRNAs. And this mechanism exists not only in C. elegans, but in many different organisms, not only animals, but also plants. Some of the seminal discoveries were in fact also made in plants, but also in humans. And this is sort of a general principle. And what was shown is that these RNAs, these double-strand RNAs, you can feed them into the worm or introduce them into the worms, and their action, this gene silencing action that they have, doesn't only stay in the tissue to which they were introduced, but it also spreads across the animal's body, the effect, the silencing, the destruction of the mRNA, and it also reaches, even when you introduce it to the soma, let's say when you feed the worms with double-strand RNA producing bacteria, they eat the bacteria, they take these double-strand RNAs, they silence genes in their gut where they eat the bacteria, but the silencing doesn't stay in the gut, but it spreads to other tissues, including also the germ cells, and leads also to silencing in the next generation. This was already in Fire and Mello's original 1998 paper about RNA discovery. So I knew that there's this mechanism, where here the double-strand RNA molecules, they breach this Weismann barrier. This idea that only what's in the germ, that soma doesn't communicate with the germline, that germline is segregated from the rest of the body, and information transfers only via the germline. This was known. I wanted to know if this also, and this, by the way, RNAi, this mechanism of silencing genes with double-strand RNA is a very common way to silence genes also for experimental purposes. We want to know what this gene does, so we just throw double-strand RNA at the cell or the organism, and it silences the gene, and we say, okay, in the absence of this gene, we see this and this effect, so we know that this gene is important for that. And it's also now the basis for drugs. There are therapies now based on RNA.
So it's again another beautiful example of, just like you said at the beginning of our chat, of like, for the audience, reaffirming that studying a worm, right? It's not just to the worm that, yes, it's 4 organisms out of 5 on the planet, but like there's implications of when we see in the worm all the way to humans and everything in between.
Right. If you're lucky, then what you study will be conserved. And many of the things, when it comes to these 8 Nobel Prizes that were awarded for C. elegans researchers, these were things that were conserved.
And and Oded, we're waiting on yours.
No, don't wait. Don't hold your breath. So they've shown that, okay? And I thought, this is a potential mechanism that could explain inheritance of traits as well. This is great, but it's the artificial double-strand RNA, and we don't know that it really transfers also natural traits across generations. And the first effect that we studied was protection against viruses. Another amazing thing in the worm, and this really attracted my attention when I just started my postdoc, is that at the time there weren't any natural viruses known to infect C. elegans. So C. elegans is remarkably resistant to viruses, and this is although it doesn't have B cells, T cells, NK cells, right?
Right. It doesn't have your usual immune memory that you think of when you think of us.
Right. The way it guards itself against viruses is using small RNAs, using RNAi. And in fact, when I was publishing my paper about transgenerational responses against viruses, that I'll explain in a second, really in parallel, just a few months before, there was finally the first virus found to infect C. elegans. Okay? But this was found in a worm that was isolated in France from a rotten apple in Orsay in France. And this C. elegans, while isolated, was found in this apple, had a virus, but it was also found to be defective in making RNA. Okay?
Aha.
So we know that, right? So we know that RNA is an antiviral mechanism in C. elegans, remarkably efficient. Now, part of the reason that it's so good to study RNA, small RNAs, transgenerational inheritance in C. elegans, is that they have a very strong RNA system. We also have RNA, humans, but C. elegans have a stronger RNA system. Not the only animal with a very strong RNA response. There are many, but C. elegans, in many cases, the reason to study a model organism is that it puts the spotlight on something. And this is true in so many cases, for everything, there's the right model organism. Flies have these big chromosomes. People could find, the first gene, and do, correlate chromosomes with Mendelian inheritance, so on in flies because they have such big chromosomes. And also first, short generation time is of, doing crosses, stuff like that. Aplysia have these big neurons, and you can study classical conditioning in them. Kendall's Nobel Prize. Certain ciliates have tons of telomeres, so they were discovered in ciliates. So many animals, put the spotlight on one particular example. One of the things that C. elegans really puts the emphasis on is RNA. Okay. So they don't have classical immunity like we think of it, but adaptive immunity, but they are protected against viruses. But at the time when I started my work, there wasn't this Orsay virus yet. All that existed back then is this artificial system for expressing viruses in C. elegans, where you have this fluorescent virus. I mean, when it is expressed, it shines in green, and you know that the worm is infected. And people before me have shown that that normally the worms destroy the virus very easily. They stay black. The virus never shines in green unless you mutate the RNA machinery, turn it off, and now the virus replicates.
Similar to that one that was eventually found in the apple, right? Same mechanism, that natural variant of the RNA mutation. That's what you had to do in the lab to get this to work.
Right. So in Orsay, they found a particular mutation. In the lab, they could study many different mutations in different genes to see how the different factors affect the pathway, but essentially it's the same. And so what I did was I took this artificial virus and I expressed it in the worm. Okay? The worm suppresses it because it has a functional RNA system. But then I use very simple genetics, crosses. These are really experiments that someone in high school can do easily. It's easy. You don't need to. I'm not a great experimentalist, setting up complicated things. This is the beauty. It's very easy. Okay. I took this worm that has the virus and it suppresses it effectively, and I crossed out and I set up a cross, and then I crossed in a mutation so that in the next generations, not in the F1, but in the F2 generation, 2 generations down the road, this worm, which was originally infected with the virus, will now produce progeny which are defective in RNA. So they can't make their own small RNAs. Okay? And what I did now is I challenged these worms with the same virus. And what we saw is that they are protected. Okay? Although they can't make their own small RNAs, they are already born with the protection.
They should not be. If everything is the same, if it's just RNAi, that one gene, because it's mutated in that, you shouldn't be seeing any protection. And yet we do.
That is unless, and this is what in fact happens, they inherit the small RNAs already made from their parents. Okay? And this is what happens. And it's more complicated than that. Okay? For a reason I'm hesitating whether I want to go into that, but okay, I'll do it. It's slightly mechanistic, okay? What you need to know in a nutshell, we don't need to go into the names of the genes and so on, is that RNA in C. elegans, this mechanism we talked about, with creation of small RNAs and silencing and so on, happens in 2 steps. There are the original small RNA molecules, which trigger the silencing. And after the silencing happens, there's the production of much more abundant secondary or amplified small RNAs, which are amplified and made over and over and over and over. And now these original small RNAs are no longer needed, okay? This is the reason that my experiments worked. The worms that inherited the effect cannot make new small RNAs that would trigger it, but they cannot initiate de novo the production of these, but they inherit the amplified ones over and over and over and over, okay?
They can keep amplifying these.
They can keep amplifying, and this depends on an amplification system, enzymes that are called RNA-dependent RNA polymerases. What they do is they make more and more small RNAs using RNA templates. And this is really one of the keys to small RNA inheritance, And in this paper, we pinpointed one of these genes called RF1 as important for amplification so this is sort of the mechanistic trick that the worm does to amplify. We talked about it that you need a mechanism to amplify the effect over and over again. Another thing that the worm has is the capacity to move RNAs between cells, silencing RNAs between cells, breaching the Weismann barrier. We don't know everything about it, but we know quite a lot. We know, and this is the discovery of Craig Hunter 25 years ago, is that there are these channels that actually, shuttle RNA from cell to cell.
And then again, you said, it can cross those barriers, so it can get from the somatic tissue to the germ tissue, to back to the somatic, and so you can all over prime this worm.
In fact, whether it goes back to the somatic cells, that's a complicated thing. It's not entirely clear to what degree does this happen. We even published a paper that we call this the reverse Weismann barrier. You can get stuff from the soma to the germline C. elegans, but back from the germline to the soma, in many cases, no. So sometimes you just establish the silencing in the germline, and this, depending on the characteristic of the genes and where it is expressed and all kinds of factors that we don't fully understand, in many cases the silencing stage is in the germline, but if you establish it in the germline, maybe it will continue to the soma. But we don't know whether the small RNAs themselves can move from the germline back to the soma. We call it the reverse Weismann barrier, and we are looking now for exceptions to this barrier that we defined as well. And I think there are exceptions, but we didn't publish it yet, but we have some clues.
That's so cool.
Yeah, so after this paper, this is the first trait that we studied that is inherited, in this case, viral immunity. We studied this using this artificial virus. It is, fluorescent and so on. Later, Craig Mellow's lab, Craig Mellow got the Nobel Prize for finding RNA together with Andrew Fire. They published a paper about a more natural virus that is also transgenerationally silenced using the same mechanism. And it is very possible that some viruses would evade this mechanism. For example, there's sort of a debate, and I think the evidence is in favor of one side, that this Orsay virus is not transgenerationally silenced, the one that was found in the rotten apple. So there could be different tricks. For example, the virus that I was studying, because of, for experimental purposes, lacked one of these genes which suppresses RNA. Okay? So it was a sort of a proof of principle it can happen. But I imagine, viruses are good at what they do, so they would also evade this. So it will be complicated. And again about luck, as I said, the worm from Hawaii, C. elegans from Hawaii, they don't have transgenerational RNA. Other worms do have it. So it's really a lot of luck about landing on the right thing, the right virus, the right worm, right assay, the timing, all kinds of things could really make the difference.
Absolutely, yeah.
So we showed that, and then later we showed that other type of environmental effects in the worm can also lead to transgenerational effects. We studied starvation. Later, we also studied the effects of temperature on the next generation. Other people studied that as well, and different environmental responses. Not all environmental changes are transgenerational inheritance. Some are, some aren't. It also sometimes dependent a lot on how you do it. Okay, so it's complicated.
And that makes sense too, right? Like, it depends how you do it, because it is environment, right, infecting phenotype. So how you're doing it, the environment in which you're doing the experiment, presumably just should make sense based on the description alone.
Right. And it's sort of, if you really want to, confuse yourself, it's a moving target.
Yeah.
Because it keeps changing. And how do you so if you take a worm, is it exactly the same as this other one that I took from some? So maybe its genetics is different, but maybe the epigenetic, heritage is different. So, for example, now that we study everything, and people didn't used to do this in C. elegans, is we keep the worms, well fed in a certain temperature for multiple generations before we start the experiments. Okay? Before that, people you see in different C. elegans labs, if they don't study epigenetic inheritance, they study something totally different. I don't know, development. So you have people with worms that are totally contaminated. They don't know, or you put them in different temperatures because you want to accelerate or decelerate development. So you play with that, not thinking about the possible epigenetic implications that would happen, because you don't study it and you think, genetics is the same. I don't have to worry about that.
It's like you're studying it like this genetic phenomena in isolation of development, independent of what might be going on with the worm. Like you're saying, if like you even said, like you're finding on temperature, right, that could be passed on to the next generation. If you're shifting stuff around for development on temperatures, you could inadvertently be influencing its epigenome.
Right. So in labs that study, the labs that study transgenerational epigenetic inheritance need to make the protocols for making this consistent and so on, and take this and this, now people do much more than before when we weren't aware.
A question from the chat too that I think is important on this isn't just a binary switch, right? This epigenome of like whether or not you're protected or not, whether a heat virus, it's a dial system, right? And that's presumably influenced by how you're setting up the experiment.
Totally correct. Yes, making things more complicated.
Yeah. We love complicated here.
I have to add that I mean, we're looking at phenotypes, viral resistance, size, all kinds of gene expression. But importantly, we can also sequence these heritable small RNAs. So we can quantify them, and we know a lot about how they are carried transgenerationally, by which proteins that bind to them, and we know how silencing is executed. So the mechanism is quite well understood at the molecular level, and many of the molecular players we know about. So, for example, one thing we can do is to better control it is not only to keep the conditions constant and monitor that, but we can also, let's say, neutralize inheritance by mutating a gene that is important for inheritance, sort of create a blank slate. Again, I have to say this is not perfect, because when you now stop inheritance, it's not that there aren't small RNAs. There are many small RNAs there. It just works differently, and they compensate for one another. So sometimes you just create a mess. So it's like this saying, you can never put your foot in the same. It's never the same. How does it? I don't know. You know how this saying. You can.
Also, I'm Hungarian, so it's also like I don't know the English, but I think I know what you mean. Like the lightning striking twice, almost in the same place. Yeah, and it's also the fascinating element too, as chat's pointing out, that knowing how long these effects last as well, right? So in your studies, like how long one of these these stressors last in terms of generation versus if you're inadvertently changing temperature for studying development, right? That could be a very different generational time lasting. So how does, accounting for that difference as well?
Right. So what we do here, I mean, people in different experiments using different environmental challenges found that many times, in most cases, the effects last for a few generations, typically 3 to 5 generations, and then they go away. And this is true at the population level. At the individual level of the worm, some effects are very long. It's very difficult to do these type of experiments to quantify the duration of the effects with complicated stresses like starvation, so on. So most of what we do, and this is a very powerful tool that we have in SciAnts, which is the key to the success of the model, is that we just feed the worms with double-strand RNA that silences a particular gene that we have a reporter for, like a fluorescent reporter. And then we just count how many generations is this gene silenced. This is a very easy thing to do. And you do this in thousands of worms. So we, for example, have a paper where we started by silencing the gene in the mother, and then we looked at progenies that were, my former student, Leah, she looked at 20,000 individual worms to sort of understand how it is segregated, the effect across generations. Why do some worms inherit it for longer or shorter? Why do some individuals inherit while others do not? Sort of trying to understand the rules of inheritance, the principles of the dynamics of the effects. And we came up with a few principles. I think it would be too technical to explain, but I mean, you can read the paper. I mean, she has 2 papers on this. To those, experts in the audience want to look. This is, so the first author's name is Leah Rechevi, and these are papers in Cell in 2016 and 2020. So you can look at these papers. But what we found there is there are principles that, based on which you can predict for how long the effect would last and how penetrant it will be. And also that there are genes that function in determining for how long and how strong the effects will be. We call these genes motek genes. Motek is Hebrew for sweetheart. People don't know that. But the acronym stands for Modified Transgenerational Epigenetic Inheritance. And in some of these motek mutants, the effects last for a very long time. So, for example, there's a gene, it's called MET2. It affects the chromatin. It's an interesting gene. And there, you silence a gene, just stays silenced forever.
So it just generation-wise just keeps going and going and going and going.
And there are other genes like that. And we published a few papers about it. There's another gene, PPTR1, which also has the same phenotype. Worms that don't have one particular type of small RNAs, this is discovery of Scott Kennedy, they are called piRNAs. And you silence the gene with just exogenous double-strand RNA, they also, silence it forever. So there are different mechanisms that time the effect and also that limit it, probably on purpose. There's probably you want to restrain it. And this is where you theorize, and we don't know this really the explanation, but it's sort of our thinking about it, that epigenetic effects for environmental triggers normally last for a few generations and then go away because that is their advantage. This is sort of a soft inheritance. If the environment changes, you no longer need this, then you go back to the hardwired, safe genome encoded program.
Right. It could almost be detrimental, right? If you permanently switched and then the environment's just transient and all of a sudden it reverts back, then you'd be the one who's no longer going to be able to successfully reproduce. And so it's a negative. It's really... Right. It's a really elegant system to be able to rapidly adapt to the environment, and it's cool that these genes exist too that can regulate that. Right. A question from the chat: why sweetheart?
Why did I choose sweetheart? I know it's also a particular type of sweetheart. But we had lots of phenotypes that we gave names that are sort of around this area. The rest we didn't publish yet. How did probably I was trying to force some word on the words that made the acronym, and or something.
I like that. But it's a memorable good name. It's a memorable good name to it.
Right. And also, the reviewers didn't understand it, so they didn't disqualify it because
perfect.
They didn't know. Yeah.
And actually, it was fun. One of the members of the community asked the question right before you were answering it too, is that when Leah was doing these crossing schemes and like the 20,000 worms, the question was, does the expression pattern, is it the same every time or is it different? And it sounds like there is this range, right, of working with the worms. And that's how you came up with these governing rules and identified some of these candidates, is because there was this spread between the genetic factors and how they were inherited.
Cool. Yes.
Yeah, it makes it complicated, but it's really cool. Like these rules that y'all set out and you established, it's almost like you're the Gregor Mendel of our time, right?
No, I was looking for beautiful ratios like Mendel, 3-1, 9-3-3-1. We didn't get that, unfortunately, but we did get some interesting principles that we can that are useful.
Do you think that the reason that there's not these like really nice ratios is just because it is a dial rather than a binary system of inheritance?
That is also one of the reasons, probably. That is probably one of the reasons. Yes.
So, what are some of the other phenotypes that y'all are looking at that can be inherited this way? And a question that we get a lot from the chat with this kind of inheritance is, how worried should I be as a person?
Okay, so two different questions. Okay, let's get with the worry let's wait for that for a second, but I'll definitely address it. Okay, first about other different phenotypes. There are many different phenotypes. What I'm very interested in, and we're going back to Cuvier and this idea of the brain directed inheritance. So what we've shown first at 2019 is that if you change the production of natural endogenous small RNAs in the brain of the worm, it affects the next generations. It is a paper Posner et al., again, to those interested, in Cell 2019. But that was an artificial system. Took the worm, we changed the production of small RNAs in their brain. It affects the next generation. Also, gene expression in the next generations, but also a particular behavior, the ability to do chemotaxis, to find food in high temperature. Now we have two new papers that were pre-printed maybe half a year ago.
We actually, Oded, we talked about the papers on stream because we saw the preprint go live, and we went over the findings of it too because it was just so cool. Like there's you have a big fan club here in this community, so every time there's a paper, we always are like, Oded has a new paper, let's take a look at it. So, but yeah, no, please explain. I just wanted to point out that we were very excited when we saw those preprints come out too.
Oh, thanks a lot. So in these two preprints, this is a story. One of them was led by my lab. The other one was led by Bjorn Schumacher's lab. Is that we've been working for 10 years, literally, about that. What we wanted to do there is to see whether it's enough that the worms would just perceive a change in the environment using their nervous system. Just think about something for this to affect the next generation. And as I told you, high temperatures generate transgenerational responses. But what we showed in this paper is that it's enough that the worms would just think that there's a change in the temperature, even when there's no change in temperature, for transgenerational effects to occur. And the way you do this is that the worm has just 302 neurons. Many of the neurons respond to temperature, but there are two main neurons that are called AFD. Every neuron has a name, which are the main thermosensory neurons of the worm. These neurons are needed for the worms to know in which temperatures they are. And the worms can move up and down temperature gradients, and they can also learn to like the odors in which the temperatures in which they are born. So if you grow them at 20 degrees Celsius and then you move them in adult to higher temperatures, they will go to the 20 to the lower temperatures. You grow them in high temperatures, and then in adulthood you move them to low temperatures. They will go to the high temperatures. Okay, so they prefer the temperature in which they were born.
And if you remove those neurons, do you lose that?
Then they don't do it. Yeah, they don't do it. And they are very sensitive. They sense very small changes in temperature. The worms cannot regulate their own body temperature like us. But they are very tuned to temperatures, and they can move to different temperatures to protect themselves. And so what we did in these papers is just manipulated the activity of these neurons. So we make the worms think that it's hot or not. And this changes silencing of a reporter gene in the germ cells for many consecutive generations.
Wow.
We did this manipulation of the neurons using 3 different ways, complementary. This is why it took us so long. We really studied it in depth, also how the signal moves from the neurons to the germline. In this case, it's not small RNAs that move from the neurons to the germline. It's a cascade of signaling molecules. Starts with a neuropeptide and changes to serotonin, so it's a very complicated thing. But here, the bottom line is that it's enough just to think for silencing to change to the next generation. Beyond Schumacher's lab, in the other paper that we published back to back, is they showed that also the genetic makeup of the germ cells changed. So when you disable these AFD neurons that sense temperature, there's more transposition, jumping of transposons in the germline, and this is and once you have a genetic change, that's permanent, that's forever. We think probably that the two effects are connected, because one of the main mechanisms that prevents jumping of transposons, these jumping genes in the genome, is small RNAs. In fact, many of the mutants where you have transposition are mutants effective in RNA. Okay, so that's what we showed in that paper, and we're studying what else can the brain transmit to the next generation, and what are the limits of this process. Another thing, and this is how I will connect to your other questions about should we be worried about this. We are studying whether this phenomena, transgenerational inheritance through RNA, happens in other organisms. It's a big part of the lab working on different animals, trying to find evidence for that.
What kind of animals, Oded? And is there, and kind of to harken back to what you were talking about earlier, is how does one choose these other animals also? So like, what are the ranges, and then how do you pick it? And then also, how do you pick what line to test, right? Because like you said, if you had tested the nematode, the C. elegans from Hawaii, we wouldn't have had this. And so it's like, if you get negative results in these other organisms, how does one even interpret that? I know that's a lot of questions, but these are very exciting things.
These are, the questions that I worry about. So a lot to unpack. First of all, organisms that are good potentially for transgenerational studies have to be amenable for experiments and long tracking of many generations, okay, and easy propagation and so on. So one of the reasons people say, maybe it happens in C. elegans because they have a short generation time and they don't go very far. So it's worth it for the parents to prepare their children for what they experience, because there's a high likelihood that they will experience the same environment. Now, my kids might live in China or something, and it would take 20 years. So maybe it's not worth it to prepare them for what I experience. Okay.
So almost the counter is this is a worm-specific thing only because of their lifestyle, and that it wouldn't apply to anything else.
Right. So I think to this I would reply that maybe, but some changes, it depends on the scale that you look at things. Some things, the scale is very long. If you think about the viruses we encounter, we encounter them or different. So I don't think that's a great argument, but it is an argument, okay? And also there's transgenerational inheritance in plants, and again, they are sessile organisms, that they don't move. So again, relevant, okay? But I don't think it has to be, this is, one thing that people say, you need to test. The other thing is that for me, it's very important to choose organisms that you can just challenge with double-strand RNA. Because as I told you, one of the strongest things that we have is the capacity to just silence a gene and then monitor its silencing across generations. When you study just complicated traits like response to stress, many things happen. Many genes change. It's hard to follow. Even when you just study normal genetics, if you're studying a complicated trait, like, height, there's so many genes involved, it's very hard to do you need to study. And so there, the genetics works only when you study, sequence hundreds of thousands of genomes. So, but if you science just one particular gene, I introduce double-strand RNA, it sciences just this gene, I'm just following it. I know exactly what to look for, sciencing of this gene. Then it's a tractable system. So I choose animals where I can start with the double-strand RNA. Later, you can find the relevant environmental conditions to study. But it's a very good starting point. And then you can do genetic screens, you can do all kinds of things. So what we talked about, and we talked a lot, so I'm summarizing, I don't know how long we've been talking, but for quite a long time. So I'm summarizing it very shortly. What we've shown is that in a very simple nematode, which is a great model for studying inheritance, neuroscience, all kinds of things. There's inheritance of, I'll say, acquired traits, inheritance of parental responses across generations, not by changes to the DNA, but because of changes in small RNAs. These are the other molecules that are carried off transgenerationally, and this is challenging many of the assumptions that we had for hundreds of years about how biology works. And we know quite a lot about the mechanism, and this allows the worms to create transgenerational protection against pathogens, to remember or be affected by the dietary states of the ancestors, and also in some cases to be affected by the activity of the brains of the ancestors. It's not because of changes in DNA, it's because of changes in RNA molecules, which are inherited and amplified across generations owing to a different mechanism, which is amplification of RNA using an amplification system that is called dependent RNA polymerases, which take RNA and just make more RNA like it.
And so it lasts across not just from parent to offspring, but to the grandchildren and the great-grandchildren. There is this maintenance of either like an inherited immunity like you're talking about, or temperature preferences, or starvation, and it's an effect that we've seen in starved people during World War II as well.
Right. So in that case, I said that this is an intergenerational effect, not necessarily requiring us to evoke really epigenetic inheritance. Very interesting effect, but it doesn't necessarily have to be the same thing. So what we were talking now is about, does this carry to other organisms? Is it just a C. elegans thing, special for this, just these particular nematodes have, or is it conserved also in other organisms? And I mentioned in the beginning, so first of all, 4 out of 5 animals on this planet is a worm. Just one important fact to know, that is, worms are very relevant for everything. So even if it was just a worm thing, it would be probably relevant. But we are, of course, interested to know whether it happens in other animals, first in other worms, right? And then in other animals in general, and finally us. Does it happen in humans? And I started saying that what we're doing now is we're examining whether this happens in other animals. And you asked me, how do we choose the animals? I said, first of all, we want an animal where we can just silence a gene by just introducing RNAs to it, double-strand RNA, and looking at the silencing of this particular gene. And many animals are not like that. Like, you can't do this in mice. So we're studying this really in many animals. I'll tell you just about one. This is something that we didn't publish yet. Okay, but we have very strong evidence that we hope to wrap up in a reasonable time, that it also happens in planaria. Planaria, flatworms, have a very long and complicated history when it comes to RNA memory that ended up with bombs sent from the Unabomber and people injured. But we have now very strong evidence that there's also RNA inheritance in planaria. Although, interestingly, so first of all, planaria and C. elegans, both are worms, but they are very, very different. Planaria are flatworms, C. elegans are roundworms. They were separated in evolution by 550 million years ago. Okay? Humans and mice are 90 million years. So this is very, very far. And they reproduce totally differently. They're just totally different.
And they also have they're stereotypically, right, studied for regeneration, right? Not even for memory inheritance, but you chop them and they regrow.
Right. In fact, they are very bad for studying genetics. Okay? Because they reproduce, when they reproduce, they don't go through the bottleneck of a sperm and an egg. And one fertilized cell is just, they reproduce by fission many times. So you have many cells carrying the information to the next generation. In fact, Morgan, maybe the person discovered the first gene in Drosophila, one of the fathers of genetics, the molecular area of genetics. He studied, before he moved to Drosophila and made the discoveries, he studied planaria, and he said, after studying planaria, that inheritance is a topic that shouldn't be studied at all because it's not scientific, it's impossible. Only when he found the right model organism that changed his mind. And then he became also a very big, it was very much against inheritance of acquired traits, and Lamarck is one of the leaders in that. Okay, but yes, planaria, they're hard to do genetics with, but they're easy to do inheritance of RNA with. You just feed them with double-strand RNA, mix it in the liver that they eat, and we see phenotypes that last for a very, very long time. After all of their cells already, were replaced multiple times, And after the original double-strand RNA that we fed to the worms is gone. We can sequence it. It's gone very fast. After 2 weeks, you have nothing pretty much. But the effects last half a year. And
Wow.
the phenotypes that we're studying are dramatic. So we're talking about, 2 heads instead of 1, loss of the eyes, very striking phenotypes, nothing that you can, nothing subtle. And another amazing thing, so this is all sort of build up for the paper that we will have eventually when we finish this, I hope, probably in a year or something, that you can also move it between individuals if you just take a small part and graft it on another animal.
Wow.
Which is amazing, because then this graft takes over the host and it also transfers the effect.
So that means that the small molecules, whatever is in that graft that you transport onto that new organism,
they can move between cells,
leaks out, right, and then hits all the other. That's incredible.
And the most incredible thing, in my opinion, is that these planaria, they don't have RNA-dependent RNA polymerases. This mechanism which amplifies small RNAs in C. elegans, they don't have it.
So does that mean it's a different molecule?
So that is the big question. And I think that probably, yes. I think there's an alternative mechanism. It's still RNAi, still small RNAs are made, but the mechanism is different.
Wow.
And this is, to me, very, very important, because it opens up the possibilities. I'm leading to humans. When we talk about humans, one of the big criticisms, people say, humans, you show that in C. elegans, it happens because of amplification of small RNAs using RNA-dependent RNA polymers. But humans don't have them. So what happens? So now we have proof that this can also happen in animals that don't have the same mechanism by other mechanisms. And there are different epigenetic mechanisms that can perpetuate gene silencing states in different animals and organisms. So for example, C. elegans, they don't have tyrosine methylation, is one mechanism for silencing genes, while humans have it. In plants, RNA inheritance is happening because of both small RNAs and DNA methylation. In C. elegans, it happens without DNA methylation. And so I think there could be all kinds of feed-forward mechanisms enabling this in other organisms. Still, that does not mean that it would happen in humans. As I said, even in some C. elegans collected from other places, it doesn't happen. We don't have good proof that this happens in humans. It's hard to study. We talked about this in C. elegans. You can study many generations fast. You have another advantage, which is two advantages for studying this in C. elegans, which is also a criterion for choosing animals you want to work with when you study this, is that in C. elegans, you can sort of neutralize the effects of genetics. The worms, if a worm, sisters that derive from the same mother, they are almost genetically alike. We're talking about humans, we are also genetically different, always. That complicates things.
Yep.
In addition, in C. elegans, you can really separate nature from nurture. You just grow them in a dish, in a petri dish with just one type of food, E. coli bacteria. You control the temperature, you control everything. When it comes to other organisms, in many cases, the environment is complex, introducing noise and complexity to the analysis. So when you choose a model organism, you wanna control for that. So my important disclaimer is saying I don't know whether this happens in humans. Time will tell. Okay, so don't worry, everything is okay. And I would also remind the audience that also this is the case in genetics. It started from experiments in peas, of course, by Mendel, later flies, bacteria, viruses. It took a while to understand how it happens in humans. And, when DNA was found to be the heritable molecule in bacteria by Avery, people said, yeah, maybe bacteria don't even have genes. No, we don't believe it. Or maybe it's different in them. Of course, also Mendel's laws don't hold in bacteria. So it's still DNA, and the larger principle that Mendel discovered, which is that the genes are particulate, holds also in bacteria. But to connect the worlds, it's complicated. It will take a while to do it in humans. I'm not even sure that we know how to design the experiment.
That seems like one of the really complicating factors in these type of experiments is not even like the concept of if it happens, but how to design it. Like that's the brilliant part about like how you first were like, well, how does the nematode feel, right? And it's like, well, how does one even get around that? But it's like, all right, you activate this neuron that responds to the stimulus, and you're just lucky that there is this neuron that you can do that to versus this whole network of neurons and other molecules. So it's like, again, right organism, right thought, right design. But when all the environmental factors are changing as well, it's like, how does one even control for that?
Right. It's not simple. Not simple. You can think of, for example, mapping, just sequencing small RNAs in sperm, isolate, one cell, track it in people that experience different things. I think that the key is we want to understand the mechanisms, be able to see whether it exists. So in cells, but it is complicated. So this is the important disclaimer. I would say, however, that maybe it's worth it to behave as if there are consequences to how our behaviors, even if they aren't.
Just on the off chance, kind of stacking the bet.
Yeah, because I mean, nothing bad would happen from being responsible for what you do.
No, that feels like it would be a good thing many people should learn.
Even if there are no biological consequences, and time will tell.
Yeah.
But I think that what we could start with, and this is it's a big operation. We're not doing it currently. Is let's say now every couple that goes to have a baby, they in many cases sequence the genome or particular genes to look for whether there are mutations that correlate with disease. You can also imagine a day where we sequence enough RNA from sperm to correlate with all kinds of phenomena and maybe disease. And the beauty of this is that unlike DNA, the RNA pool is changing dynamically in response to the environment. So there's something you can do about it.
Right. It also puts a lot of weight then onto like potential downstream ethical questions too, right? Of like, what is it that is good to modify? What is it that you don't want to modify? What is a gene pool diversity that you want to keep? What is it that you don't want to keep? And like, even if you could modify it, what is the outcome long term for that human patient's life, right? It's like a lot of complicating factors coming on in.
So we see time and time again that the biology, science in general, is ahead of the ethics. So we have to deal with it later. This is like what we're experiencing now with AI, for example. When we have to think about what we do with it. I also have I'm also working now on all kinds of AI related things. So I'm thinking about that as well.
If you wouldn't mind touching a little bit on that too, Oded, we get a lot of questions in the community of like, how does one even decide to use AI in scientific experiments? Because a lot of times it's like almost like with machine learning models, it's like if you have garbage data going in, you're going to have garbage data coming out. So like when you're designing AI or using it in your experiment, how do you know this is something I should test? This is something I should use. This is worth using, and I'm comfortable pursuing this down the line.
Sure. So, of course, there are many concerns about AI. One of them is these hallucinations. Can you trust it to when it just invents things that are not grounded?
Mm-hmm.
And so what I am using AI for is to get feedback and not to generate. So I'm not using AI to generate hypotheses or to, replace me.
Mm-hmm.
I'm using AI to get feedback on my experiments and papers. Have the AI look for gaps in your logic or your experiments and things that are wrong with it, and give you the feedback, and now you have to deal with it. And also making sure that it does so and it gets references and it explains itself. And you, as the expert on your topic, know whether it makes sense or not. And you can also just check it. You can decide also to ignore it, but the power is in your hands. So it's not AI to generate the science, it's AI to give you feedback about your science.
That's a really cool way of explaining it too. It's like the gaps in the logical flow of how you're almost, it's like as Chat mentioned, an AI reviewer. Here is this logical leap that you make. What's the connection here? And you have to build something in now.
Exactly. So that's exactly how we're trying to build a critical AI that will give you feedback. And in fact, you can check it out. It's something that we just launched. We didn't advertise it. We're not quitting it. We just started it very recently. I mean, I've been working for close to a year, but it's already operational, so you can use it. Okay. If you just write QED science, okay, I can also, I don't know if you can just put it in the chat or.
Yep.
Or
yeah. We've actually been populating all the papers that you've been mentioning. We've just been putting it into the chat as well.
Yeah. So I'm just I'm putting it in the chat. So QED science, I put it now. Okay. So this is a tool that I'm creating for it. It's a AI reviewer, but it's not a reviewer. I mean, people have been discussing AI review now. They're really worried about it because they're afraid that it will be that people will just do reviews on ChatGPT or something, or the journals will just use something that they built for their own purposes against your paper. Here, the idea is that it will be an author-centered tool. This is something I'm giving the authors. It's also free because you can use it. And you just give it to the authors. The idea is that you improve your own science. You just throw in a paper. Soon you can also throw in just fractional pieces of data, just a gel, a picture of. But currently it's or a presentation as you want to present in lab meetings. Just throw it in. But currently it is ready for manuscript. You put in a manuscript and it gives you a review. And the review is not, it doesn't look like a review of a human. So what you'll see if you use it, it's very nice, is that it breaks your paper into claims. What did you claim in your paper? And it builds a logical tree from your claims. Okay, and then it says, okay, for some claims, I'm finding gaps, I'm finding problems. And then you have the logic of what did you show, what is the gaps that we found, and also it suggests ways of bridging the gap, either by doing experiments, it suggests experiments, small experiments and big experiments, and also suggests textual edits to your paper to bridge the gap. For example, if I said, if I called my paper transgenerational inheritance of neuronal activity, it would say, I would add in C elegans, okay, to be more accurate, okay? And it will explain it. For example, this is just an example, okay? And what we did, we broke down the reviewer's task into many, many different tasks, and we addressed them. We keep also doing that. And so you can get this review, improve your paper, and very soon you see also, so in the website you have this a blueprint. This is what I just described, and you also have a tab that is called bottom line. And this is sort of a report that you can generate, which is brief, and it summarizes what you find and the potential problems. And you can also, comment and say, this is not a problem and then it will show up. So it's also doing the response to the reviewers. And then what I want, this is my dream, is that this bottom line report, that you will be able to just post it together with your preprint online. And then, it's a sign of transparency, and also an editor will look at it and we say, maybe I don't need the human reviewer. Maybe I don't need to wait for free. I can just use this AI thing, which, the authors can't mess with, but it gives you additional information, and it's unbiased and it's really detailed and transparent. And I can use this to make decisions because, the journals really struggle with finding good reviewers, and good reviewers have emotions, sometimes, conflicting interests, sometimes it's a lot of work, so there are many problems with review, as everyone knows. Here you can complement human review with all of its faults, but also some advantages with this machine-based review, and maybe it would speed up publication, also make it more, democratized, make it more fast and efficient and fair.
And especially co-posting it with a preprint, right? Versus, so instead of just having a preprint that's just up without any evaluation, you'd have an evaluation associated with it.
Right. And the way I want to do it, and I'm in discussions with the preprint servers and also with a few journals, is that it's all in your hands. It's not that everyone gets it. If you choose to do it, you have a report, you feel confident of this is my report, then you post it together with your preprint. And then you can transfer it to the journals. You can also send it with your cover letter or however you want. The preprint servers, they will also have the capacity to just transfer it if you agree, and then maybe it will just speed up publication. You don't need to wait now for half a year for free reviewers and be at their mercy, because maybe you already have this to help you publish.
It's such a cool. It's really, really exciting. We had a question from chat of, and I don't know if you can go into the details of this, of how did you choose a training data set? So how do you? That seems like it's not subjective of logical flow, but maybe there could be on how you train, what is good logical flow? How does that get introduced to the model?
Right. So great question, of course. So first of all, it's about you have to have many humans in the loop, and we already have more than 1,000 reviewers tagging for us, improving the models. Okay. You also have to break down the process into really create a good description of what you want it to do, and you also have to train it on real data. And by the way, importantly, everything that you upload there is totally secured and private, and we're not showing it to anyone. Okay, so this is important to know. One thing we're training it on is all the open reviews out there, and there you also have to distinguish the good ones from the bad ones.
Yeah.
Also, work is in progress as they improve, as the authors change them, and we see how they improve. So this is type of training that goes into it.
Yeah, it's a very elegant system, like you said, and just the numbers too. If you have that many people involved, that'll drown out any noise and hopefully standardize it in a way that it can be utilized.
Right. And as you'll see now, and if you upload the paper, which is out of scope, because currently we are specializing mostly in biology and molecular experimental biology. Okay? So if you upload a paper about, I don't know, zoology papers about elephants in the savannah, you'll get a sort of a disclaimer saying we're not good at that yet. Try it, okay? But with a grain of salt, we will get to that. But we are gradually expanding to other things.
That's so exciting.
So you get a disclaimer. But the beauty of it, it's not. I like fast system. Even C. elegans, it's fast. 3 generations, a generation, 3 days. But when it comes to computers, it's much faster. So that's the improvement occurs very, very fast. And please test it. I mean, I think it's very impressive already. I'm using it now for everything. All the papers in the lab, they go into that. I constantly send comments to the different authors in the team about things that we need to do, including just textual changes, but also experiments. It's smart.
That's really we're actually
I don't know if it's smart is the right word, but it's good.
Once we complete once we can finish our chat, we're going to try it out on stream as well and show people how it works, because that sounds really, really exciting. Oded, we want to be respectful of your time. But would you be able to stay on with us a little bit longer, talk a little bit about your work on the Dead Sea Scrolls?
Sure. I'm really enjoying it, so my pleasure. Thanks.
We could keep you on for hours. The questions keep coming in, and everyone just loves it's all positive feedback, and people are so excited to have you here. So thank you again for your time.
Thank you very much. Thank you.
We actually, when we started the stream today, I listed out a set of things that are topics, and I asked the community, I'm like, what do you think our guest is not an expert in? And so I have these worms and transgenerational stuff and neurons, and I put in Dead Sea Scrolls and monkeys, and everyone's like, oh, it's the Dead Sea Scrolls, it has to be, right? It sticks out like a thumb. So I think one person picked a monkey.
So I'm definitely not an expert in monkeys.
So how did you get to archaeological data and looking at Dead Sea Scrolls, which has important religious, historical context, and very, very different from those C. elegans?
So I like to do many different things. We also studied, as you mentioned, brain parasites, economic irrationality, all kinds of weird things. The Dead Sea Scrolls is probably my favorite off topic. And this happened again by coincidence. What happened, I just started a lab that was in 2012. And in university there was a retreat, 2 days retreat for all the new faculty. And in my university, there was everything. So there are people studying science, but also people studying art, everything. And in the evening, they took us to a restaurant. And on the way there on the bus, 20-minute ride, I sat next to a guy, Noah Mizrahi, who back then he was in Tel Aviv University. Later he moved to Jerusalem University to be the head of the biblical studies department. Okay? He's a biblical scholar, and we talked. He's a very nice guy. We're good friends now, and he is a brilliant guy. If you talk about being a scholar, he speaks 12 languages and knows the entire Bible by heart. It's really amazing. And it's interesting stories. If you want to bring him on the podcast, really, it's.
If you wouldn't mind making the connection for us, that would be incredible.
I'd be very happy. This guy is amazing. Okay, and so he told me about what he's doing. I told him about what we're doing, worms, and he was telling me a little bit about the Dead Sea Scrolls. The Dead Sea Scrolls are the most important archaeological finding in Israel. And Israel, is sitting in a very important place for history, so many discoveries here. But still, I think that the Dead Sea Scrolls are probably the biggest. And these are scrolls that were found in the desert near the Dead Sea, in a place called Qumran, in the 40s and 50s, late 40s and 50s. And the story is that they were found by a Bedouin shepherd who had a sheep run into one of the caves there, quite high. It's not easy to get to. And he wanted to scare the sheep out of the cave. So he threw stones into the cave. And when he did that, he heard the noise of vessels breaking. So he climbed over there, which is not easy. And he found vessels filled with scrolls. And these scrolls were found to contain the most ancient Bible. The next one is 800 years older. And also many, many texts. So they found over the years in Qumran, but also in related places around the Dead Sea, 25,000 pieces of scrolls.
Wow.
And since then, they've been trying to assemble them together so they understand what's written in them. It's a very big task because they are 2,000 years old, okay? And written over a course of a period of 300 years. And in many cases, there are pieces missing or in bad shape. Amazingly, we can read them. So they're in Hebrew. That's also amazing.
Yeah.
And it's like trying to assemble an unknown number of puzzles with unknown number of pieces with many are missing. Nevertheless, they did it. they assembled many of them and there's a big museum where many of them are found. Not all of them, not all of them, but many of them are found. It's also a whole story, like in Indiana Jones story of how they were later collected. In fact, Indiana Jones is named after Wendell Jones, which was a guy who looked for a treasure in the scrolls. And if you do a Wikipedia of him, he has the same hat and everything. And one of the scrolls is actually a scroll that is found in Jordan, not in Israel. And this is a scroll which is made of copper, and it's really an inventory of a treasure. And the scholars, they argue whether it's a real treasure or a metaphorical treasure. Noam thinks it's a real treasure because they know how they wrote prose, and this is just an inventory of this and this amount of silver, this and this amount of gold. So many people were looking for that, but no one found it. It could be buried under any garden in Jerusalem. Everywhere you dig there, you find something. But most of the scrolls are not made from copper. They are made from animal skin, from parchment. Okay, so the idea that we had is to use that to piece together the scroll pieces. Of course, people use every clue that they could. If it's a text of the Bible, we know it. We can try to piece it. In many cases, there are different interpretations. This piece can fit together with this piece or that piece, and depending how you piece them, you get different interpretation.
And it's difficult too, right? Because if the most recent one beyond the Dead Sea Scrolls is 800 years old, if you're trying to compare texts, there could be changes and parts missing
Absolutely.
or shifted or what have you. So yeah, there's an even bigger puzzle.
And there are different versions of the Bible, even within the collection of the corpus of the Dead Sea Scrolls. So you're totally right.
Yeah.
So what we did is we took advantage of the fact that they are made of animal skin, and from each piece we sequenced the ancient DNA of the animal from which it was made.
Wow.
Okay? And we first cataloged them based on animal type, so sheep, goat, cow, and so on. But most of them were made out of sheep, the one that we sampled. So then we went on to look at genetic differences between the sheep, and we said this piece should fit with that piece and not with that piece. They're all made out of sheep, but they are more closely related genetically, or maybe they are made from the same breed of sheep, or maybe even they have the same SNPs, single nucleotide polymorphism that allows you to connect them with. And we made some connections, and we also made some separations of some connections that were debated, and Noam gave us the context of new findings about the history and the scrolls.
So for a question from the chat too, is how difficult was this? Because like the half-life of DNA, right, is like 520 odd years. So you've got some degradation happening, and presumably how the scrolls were stored too. So how did you overcome the barrier, or was that a barrier really, of DNA degradation?
So technically, it's extremely difficult technically. This is why we also collaborate with the lab, ancient DNA lab in Uppsala University, Mattias Jacobsson. And my lab manager, Sarit, spent many months there in the cold in Sweden, doing the extractions and all of that. But a few things about that. So first of all, the fact that DNA degrades, when you do ancient DNA, you look for the degradation patterns. There's damage patterns that are typical. They have rules, and you want to make sure that you're looking at ancient DNA, not modern DNA. So you're looking for pieces of DNA that have these damage patterns. Okay? This is a huge problem when you're studying ancient humans. You want to make sure you're not sequencing yourself.
Right, yeah.
Okay. It's less of a problem, when you're studying sheep, because I don't have sheep in my lab, but I still have, whether the people might have, so it's always...
The clothing can be contaminating. Oh.
Yeah, absolutely. So you need to be, so everything is done in clean rooms, astronaut rooms. You have to deal with contaminations, and you can only rely on sequences that have damage patterns. When it comes to the ancient DNA field, they analyze samples that are much older than 2,000 years. Okay, when you look at about ancient humans, they go further than that. But the difficulty here was that we can't just take these precious treasures, these pieces, and just chop them in the blender and sequence them.
Yeah.
Because they're in museum and we can't even touch them. The people in the museums, what they do is they just scrape a little bit of scroll dust from the uninscribed side of the scroll. And this is what we sequence. So we sequence extremely deeply, billions of DNA sequences to get the depth that we needed, and still it's shallow. So there are bioinformatics challenges, the quality of the samples. So these are all big challenges. We also had to prove first that to the Israeli antiquity authorities that we can do this before we go and damage the scrolls. So what we did in the beginning, to convince them, and I met with ministers, I did a lot of work to get the samples. But in the beginning when people have been trying to assemble the skulls since when they were found 70 years ago, and you have pictures in the museum of them, putting all the pieces on long tables, trying to piece them together, and you see there that they work without gloves and they smoke, so you have contamination of all kinds.
No.
And another thing so you have to account for human contamination, because also we were hoping maybe to find some human DNA, but you can never say that this is ancient humans and not modern humans. So we didn't do that. So we said we can't in the paper. We say we can't. But it's a great paper. I mean, to read. I really recommend people to read it because it's a fun paper. Okay? So you can look it up. It's Anava et al. 2020. So it's my favorite paper. You see also it's written as a sort of combination of history and biology and all kinds of things. So another problem was that back then in the early days, when they found pieces that match, they just glued them together with duct tape. Okay. Sometimes they even used hot glue and glued it to perspex to this glass, and then it's dead. But what we did for 40 years or so, they've been removing the duct tape. Okay? And when they remove the duct tape, they keep the duct tape next to the piece. So to prove them first that we can get some proper sequencing, we just sequence from the duct tape, and then we move to just scroll dust, and then we move to pieces without text that are less important. And only then we went and studied the real scrolls. We also had, some fake scrolls made from parchment using the old technique made for us to experiment with. So we did all of that, sort of as a way to get, and it took us 8 years.
And that's all because of the historical context of how important these scrolls are, that you can't just be going in and testing anything. So you had to test a method that it could work on parchment. It had to be that you could work on the old parchment, that you could work on, again, these very minuscule amounts of information too. Like this dust, presumably, it's not like if I take a fruit fly brain and grind it up, that's probably going to have a lot more information in it than just some of this dust.
Absolutely. And you also have lots of contaminations from humans that touch them, maybe in antiquity, certainly in modernity, and you have bat feces from the caves. You have all kinds of stuff, that you have to deal with. So it's also heavy on the bioinformatics that you need to.
And you were saying that the transport, even from the caves to the museum, and now that you've got the people touching in the museum, taping and smoking, it's a nightmare.
No, and also when they were found originally, so they were sold to the Israel Antiquities authorities, but really people who sort of stole them, and sometimes hid them under their floors, and then wash their floors, and this damaged the. So it was a whole thing. And recently, and there are also many cases of forgery. Recently, 5 pieces of scrolls were sent, small scrolls, were sold to the Bible Museum in the U.S. for millions, and later found to be forged. And the cheaters, they are very sophisticated. Sometimes they find actual old scrolls and write on them. So you have many challenges.
So if you tested the scroll itself for age without the text, you'd be getting the right age.
Exactly. So, and people do carbon dating, all kinds of things like that. But some of them are very sophisticated in the way that they do it.
Yeah. Ooh. Wow. It's Oded. It sounds like a nightmare. It sounds like a dream experiment because it is the pulling together of all these incredible fields. But it sounds like a nightmare from a technical perspective.
It's a technical nightmare. Of course, we can't do things that we do with C. elegans, but we brought in many techniques. There are, 20 or 30 people on the paper. We did a lot of work for 8 years, and we got to a certain level. And there's more room to more things we can do. But we reached a level where some things we can say with confidence.
So what were some of the big takeaways, and what changed in our knowledge of the Dead Sea Scrolls? How did the piecing together work? And what, I guess also, I know I'm asking tons of questions. I apologize. It's just so exciting. What are going to be next step for the Dead Sea Scrolls with this project? Where can we take this further too?
So what we did is Noam gave us the context, specific pieces where there's debate, whether they fit together or apart. And in addition, there were also questions about the overall collection. Okay, so I won't go into the specifics now of the piecing together. That's diving deep into Bible and stuff like that. Okay, but let's talk about the collection for a second. As I said, this is the biggest collection of ancient texts that we have from this time. 2,000 years ago, year zero, important for many religions. Jesus might have been there. Judaism was formalized. Very important. So, and the question is, one of the questions, there are many questions, is how relevant is even the collection for the whole thing? Okay, imagine that, so the scrolls were found in a cave near the Dead Sea, which is the hardest place to live in the world, pretty much. It's 40 degrees, there's no water, it's tough, okay? There's even a debate on who was living there. So, but these are tough people, and we know about them a lot from what is written in the scrolls about their the customs and what the code of conduct. They're extremists. These are extreme people. And so imagine that now, let's take some country that is not Israel, so won't get into politics, but let's say some country, I don't know, Switzerland, okay? Imagine that Switzerland explodes and all that is left out of Switzerland is just the library of someone living in a cave, okay, away from civilization. How relevant would this person's library be to the library of someone living in Zurich or Basel? It's unclear, okay?
Right, that's a really good point. Like, is there something weird about this individual that they're living in like a cave, right, and like the worst part, like difficult. Right.
Yeah. And out of this library, out of this collection of the Dead Sea Scrolls, we extrapolate to say a lot of things about the culture and Western civilization. So many things start from the scroll. The idea of good versus evil, light versus dark, it all goes to the scroll. So this is fundamental for Western civilization. Okay.
Yeah.
And we extrapolate from these isolated caves to the broad land at the time. So how relevant was it? Okay. How do you even address this question? So what we did here is we had for example, we did a few things, but for example, the collection contains the Bible, okay, but it also which is known from other places, but it also contains some collections of texts that are very typical for Qumran, for that place. One of them is called the Songs of the Sabbath Sacrifice, okay? And this is like typical of this cult or people living there. So we analyzed pieces from this collection, and we found and we compare them. There's one of there was also these songs were also found in other places. For example, Masada, which is a different place that was later. It was also dated later. This was the last stronghold when the Jews escaped. So where they famously all killed themselves. This is another story. Okay? A parchment from these songs was also found there. And then the question is, did it get there because it was brought there from people who escaped Qumran and got there, or was it just, or did it get there independently, which suggests that it might be, the song was more known around the land, not just in Qumran. Okay?
Right. Is it something just specific to that location? Is it more, yep.
Right. Did it get there from Qumran, from where the scrolls were found? Or did it get there in another way, which suggests that then, it's more relevant because probably common all over, not just in Qumran. And we found that the animals that were used to make all the scrolls in Qumran are different from the animals that were used to make this song in Metzada. So just a different breed of animals. Okay? Suggesting it was more. So the texts are really relevant for the entire land. We had other evidence supporting this. So this is sort of a meta, takeaway on the entire corpus.
Just how cool is it that you can identify the animal on which it was written and then do the analysis of two like that's just an incredible bit of history, biology, technology. Like, it's just such a cool story.
Thanks a lot. Thank you.
Yeah, so it's that makes sense of like the tying together of those different locations. So it's like, were you able to then piece together some of those components that we didn't know that actually tied together, or that were questionable whether or not they tied together based off something like that?
Yes, so we have some piecing together, also a particular piece, and piecing apart of pieces.
And so we'll know too, like, presumably where there's gaps based on this kind of information too, like page gaps.
Mostly when there were debates of is this piece, does it fit here or here? Mm-hmm. Depending on how you piece it, the meaning changes.
Yeah. What was your did you have any component that you were the most surprised by, if there was a meaning change or just something that was exciting or cool?
I'll tell you what I wanted. It didn't happen. Probably I shouldn't say it, but I was really hoping, we'll find some pig or something, but we didn't, because that would be super controversial. But no pig skin was found to be used. But otherwise, I would probably, never allowed to publish it or something. But
it's so when you're testing, are you testing against, I guess, like a wide data bank of different types of animals onto which had been written? And that's why you even went in and tested the pig as well.
Yes. So we did that. We compared to some animals that were suspected. We also had not only parchment, but we also had sandals that were connected from disease, garments, all kinds of things that were water skins. So all kinds of things from the area, from different locations also, not only Qumran, but other places around the Dead Sea. And we studied, and
those are almost controls in a way, right? Like, yeah.
So this paper is beautiful for the controls. We have tons of controls, and it goes back to this QED science thing. What it suggests is tons of controls. Just throws at you tons of controls. I think the controls are the most beautiful thing in doing experiments.
And in this one, I love the controls, Oded, because they're so creative, right? Because like, what are you gonna do with a control if you're just looking at this as like just as a worm scientist? Like, I don't know what it's gonna be, but you, sir, like you have that experience in the art. In the history, all the experts on this paper, like I think is just a beautiful coming together of all these fields to make something really incredible and to have this really big takeaway discovery.
Thanks a lot.
Yeah, it's people in the chat are wondering too, like when the memoirs are gonna come out about just even this story alone of just like the immense challenges, you know, that y'all had to face to be able to put something like this together.
Thanks a lot. Let me tell you about one more thing. Maybe we talk about and then maybe we'll finish about the toxoplasma and the brain parasites. You want to talk about that for a second? We have time.
We have time as long as you want to be here, Oded. If you have time for us, we would like to keep you.
Okay, thanks a lot. So let's just do that. The last thing, because I think it's a cool paper. We published it last year. What we studied there, it's also very different. There is this famous parasite. It's called Toxoplasma gondii, famous for being the cat parasite, or it's really a symbiont of the cat. But what's special about it that it can only replicate sexually in the gut of felines, including cats, and it doesn't do anything bad to the cat. But if it infects a mouse, it travels to its brain and changes their behavior. And these mice are probably not, at the best shape at that point. But one phenotype is that they lose their fear of cats, and then they run towards the cat. The cat eats them. And then the parasite can again replicate in the cat. So it's good for everyone in this trio except the mouse, this deal. But aside from that, it also infects humans. It infects many animals with warm blood. And the estimates are that it infects a third of the human population. Most of the time when it infects us, we are fine. It is dangerous to people who are immunocompromised or to fetuses that don't have their immune system developed yet, fully developed yet. This is why,
in terms of dangerous, you don't mean that they're going to run towards a cat.
No, I don't. I definitely don't mean that. It means that they will go to the brain and damage it.
Gotcha. So it's going to be to both immunocompromised and that's why for the infants, there's always the thing that pregnant women shouldn't be like handling cat litter for that reason too.
Exactly. For that reason, and also pregnant women do tests for Toxoplasma antibodies, and there are also good drugs to treat Toxoplasma. Okay. So we can handle it, but it's relatively safe because if you're immunocompetent, then many times you won't even know that you're infected. And in older people, the chances so it's a third of the world population, but in some countries, in older people, it gets to 70 or 90% of people have it, depending on the country.
Wow.
Okay, so it's very common. Most people studying Toxoplasma, of course, try to get rid of it. What we thought is that we can maybe use it for our advantage because, of course, there is the blood-brain barrier preventing the delivery of drugs to the brain. And drugs needs to be small and have certain chemical properties to pass the blood-brain barrier. Then our neurons are surrounded by other cells that create a barrier for most molecules. So proteins, for example, cannot just swim from the bloodstream and reach the brain.
But the toxoplasmosa can cross our blood-brain barrier.
Right. The toxoplasma has mechanisms to also, it hitchhikes on immune cells and it can also move on its own. It gets to the brain. It infects in the beginning, it infects other cells, but it gets cleaned from the rest of the body by the immune system. And it stays in the brain, which is also immunoprivileged. And once it's inside your neurons, it will stay there forever.
So if I have it, I'm not getting cured.
No. But that's probably not so bad most of the time. So what we thought is that it's very convenient it's very easy to genetically engineer it. So what we did is when it reaches the brain, to manipulate the cells of the brain and to evade the immune system, it secretes proteins into neurons, and it has 3 different secretion systems that it uses to secrete proteins to the brain. One of them is called micronins. It secretes proteins outside of the cell. The other one is called the rhodopteres, which is like a harpoon that it shoots into the cell to get into the cell. So it's a transient thing. And the last one is called dense vesicles. Once it's already inside the cytoplasm of a cell, it creates this vesicle that surrounds it, vacuole, and then it secretes these dense granules into the cytoplasm forever, constitutively. What we did is we took proteins that the toxoplasma normally secretes from the different systems. We focused on 2, on the rhodopteres and the dense granules, and we fused them to proteins that have potential to be targets, to be drugs, so they have therapeutic potential. And then they just hitchhike on the toxoplasma proteins and get secreted from the different systems. With many types of proteins tested which can be delivered, which cannot be delivered. And we saw that the toxoplasma is quite efficient in delivering proteins to the brain, passing the blood-brain barrier and delivering proteins. And the proteins also in certain cases remain active. So for example, we deliver the protein that a Cre recombinase delivered into the cytoplasm, but then to function, it needs to go to the nucleus where it operates on the DNA. The one that we use, the Cre it removes the stop cassette from the genome, which makes the neurons now express a GFP that shines in green. So you can know that it was delivered to the neuron, and it was also, it remained functional.
Yeah.
And then we did it in cells, in stem cells, in human brain organoids, and also in live mice where their brain shines in green.
Wow.
We use the chemical procedure for making the brains of the mice transparent, and then you can quantify exactly where this worked.
Wow. So how well does it work? Does it work in a little zone? Does it work in a big zone?
So it gets delivered all across the brain. We don't have the way now to, just target a particular brain area, okay? And the efficiency is relatively low. So these are things to improve, and we have many ideas on how to improve them. But it can do things that you can't do in other ways. For example, it can deliver very big proteins. The alternative is gene therapy, but gene therapy is limited in the cargo that you can put into the virus. Here you can deliver very big proteins. And also when gene therapy started, that was a very long time ago, the efficiency was extremely low. Over decades, it was improved, and now it's already, in use, curing people. In our case, it's just a proof of principle. There's lots of work to do, lots of obstacles to overcome. For example, you need to make sure that you tame the parasite so it won't be dangerous. There are also ideas on how to do that. You have to improve the efficiency. It's by no means ready for the clinic. It's just a proof of principle, but I think it's another way to deliver proteins in the brain, and we are missing such ways.
So this would be like almost you're doing a drug screen in a way where you've started now with a molecule that can do this delivery mechanism in the toxo, and now you're going to modify it to make it more efficient and better. And then once you're going to further modify it and go through iterations, right? Safety, efficiency, until it's ready for something clinical, just like you would for a small molecule.
You modify the molecule, and you also modify the parasite. So for example, you can strip it of immunity, of pathogenicity genes. Okay, you can put in it a death cassette, so it will kill itself after a while. You can make it addicted to something and then stop giving it to him, so it won't be dangerous. So you can change the promoters to make the delivery, the expression of the gene of the protein that you want to deliver stronger. All kinds of things you can do. Lots of work. It will take a long time. Should be a community effort. It's not something we can do on our own. And again, I'm not alone on this paper. I have collaborators in Europe, in the US. But we're just seeding the seed, and now we need to do the work.
Are there particular drugs that you would, or the team would be most excited about being able to test? Like, are there drugs nowadays that maybe get to the brain here and there, but it would be so much better if it could be more efficient in how it could be delivered, that could make a big difference to people's lives?
So we looked at a very big list of proteins that have the potential to be beneficial, and there were some criteria that they had to address. For example, we know that we cannot deliver it to the entire brain in very high levels. So it needs to be a disease where a particular protein is missing that you can restore just in small amounts to a particular brain part, and it will help. For example, if you just start something and then, there are secondary effects. It also needs to be diseases where there are just a limited number of proteins defective. In some diseases, there's lots of things going wrong. It will be hard to correct. So we chose, for example, one of the conditions that we started with is Rett syndrome, which is just, about with one protein missing that you need to deliver in low amounts. Okay. And also, it needs to be a disease where it's not too late to intervene.
Right.
If there's a brain disease and there's accumulative damage, it starts very early, it's a developmental thing, then it would be hard to intervene later in life. So there are all these criteria that guide us in choosing the right conditions and proteins.
Yeah, it's such an elegant system, though. It's so beautiful. So one question we have from the chat is, how did you come up with this idea? Because it's brilliant. And so it's like, for these kinds of ideas, like, what is your inspiration for coming up with this kind of therapeutic? Because again, it's, as you're explaining it to us, and I think that's part of, because you're very eloquent in how you explain it, it makes sense. But how do you get these ideas? How do you, like, we watched the video when you got awarded at Tel Aviv University, a really big award for young innovating scientists, and you're with your daughter in the park and having coffee, right? And saying that just like, in my dreams, I'm just coming up with it. Where are these ideas coming from? Because it's just, again, it's so brilliant and so cool.
So I think that first of all, there's a saying from, Leonard Cohen, who said, if I knew where inspiration come from, I would go there more often. We don't know. From everything, from movies, from films, from conversation, from everyone. I think as a strategy, I don't get all my ideas from that, but sometimes as a strategy, what I do is, you can think of how I will fix something or how I will come up with a good idea. That's hard for me. What I do sometimes, and it goes back to the very beginning of our conversation, is I say, everyone thinks this can't happen. Let's try to do everyone, or everyone thinks this happens in a particular way. Let's find an exception. Sort of try to challenge dogmas. And then, if you find an exception, they will be important because the dogma is so fundamental. It's the same with the toxoplasma. Everyone is trying to get rid of the parasite. Let's use it for our advantage. Everyone is saying, you can't inherit acquired traits. Let's try to find. And in biology, the nice thing is that there are always exceptions. There's exceptions. It's arguable whether there are even rules.
Right?
Real laws. Like in physics. I don't know. Even in physics, some people argue, but in biology, there are definitely always exceptions. And it's frustrating on the one hand, but on the other hand, it's a great source for coming up with ideas. Just take a very important something, study its history and try to find exceptions. So this is sort of a like, if there's something systematic about the way I work, then that's that.
I absolutely love that. One comment that we had, Oded, from the community, and this is, I think, a nice way to round out the discussion too, is people have been extremely impressed by your science communication skills, not just today, but just in general. Like, we had folks who knew you in this community, who knew you from 2019 from following your Twitter, right? So like, and they're just like, oh, I know this guy because I follow him and he makes things accessible, and like the memes and the GIFs and going on Joe Rogan and doing TED Talks. It is clear that you are extremely passionate about what you do. Why do you feel that it's so important to disseminate this information that you're studying? Because it's, again, you not only make it accessible to scientists, you make it accessible to everyone. And so why to you is that important?
Well, I think that, first of all, I cannot resist myself. I think that we as scientists should take ourselves lightly. I think when we are pompous and too serious, it also limits us. If you're too serious and you cannot challenge yourself and you cannot try things that are out of your areas of expertise. So I think it's very important not to take yourself too seriously. I also think that, many of the jokes I do on Twitter, for example, or Blue Sky now, and so many of them are not about the science, it's about the scientific process. So, for example, the inspiration for, you see that I create this QED science review thing. You see that many of my jokes for years are about science publication and reviews, and all of the bureaucracy in science and the thing that scientists connect with, because this is what we deal with. And many of it is very arbitrary and just, idiotic. So we have to laugh at it to cope. I think laughing about things is just a coping mechanism. And it's a little bit like science also. You look at something from different angles. It's like doing science or coming up with experiments and controls. And so it's the same skill pretty much. I think it's exactly like the skill of scientific inquiry. I think it's very much connected to humor. And my social media accounts are mostly for scientists. I don't do too many jokes that are outside of science, but so for me, it's part of working, really, part of coping. I think that if you allow it in your team and with other people, then they are more creative. You want them to challenge you. You want them to also make fun of you. That's fine. So this is what I do there in psychomedication. The other things that I do, which is not necessarily about humor, is just I want to make science fun because I'm just much better when I'm having fun. I think everyone pretty much. Everyone is better when they are enjoying what they do.
And that's the same idea that we have for this stream and show as well, is that it's important to have fun with it, right? It can't just be this dry presentation. Like if I don't see your passion about it, right, I'm not going to be passionate about it. But that's what our community is talking to in the chat, is how everyone in this 800 plus people that have been here today have seen in your eyes just the love of what you do and that excitement and that joy, and that it's just, it comes across so well and so easily. It's just so much fun to be able to communicate about this. And I feel like that's, I think, really important, as you're pointing out, for being able to do quality science outreach and getting people excited about science. You have to be excited about it too.
Right. And you have to also make it fun for your team and for your community. So for example, with this Woodstock of biology meetings, we're trying to rethink how conferences should be. And you see what it brings out of people, the creativity, they dare. So it's just, they talk about new things. So it's just, we have so much potential. I think really being a scientist, we complain a lot, we make jokes, but in the end, it's the best job in the world. I really do think it's a loophole in the world. We have unparalleled freedom to do whatever we want, to work in our own time. We are always surrounded by young, creative, smart people. The things that we do don't need to be useful. It's just it's enough that you're just enjoying and it's interesting to you. It's really who does that? It's amazing. But still, as a scientific community, and part of it is just a historical accident, we forced on ourselves certain structures that are very limiting. The way we present, PhD has to be this, so many things. And the world is changing, and we need to also change academia and how science is done in academia and outside of academia. So this is why I also created this QED thing, and for me it's as important as doing the science. I think, for example, if my efforts contribute to changing science publication, maybe it's much more important than the science because it's so limiting.
And it's the same too, I think, for accessibility to the science as well. Like, if I think more scientists were willing to discuss their work in the way that you are, right? I'm not just going to be up here talking technical jargon in a suit, and not make it accessible, but like the way that we've woven the conversation today, going from the broad scale to the small molecule and right back, I feel like it can be accessible to people who are non-experts, to people who are just interested in the topic. And I feel like that's Absolutely. A really important skill.
Absolutely. And they have a lot to contribute. They have ideas. When you lose the jargon and you explain to them, they will bring new ideas, new original ideas. And, in many cases, of course, everyone knows that the really new ideas come from people who are outsiders. So it's not only science communication. Sometimes people think about, you need to explain to people out there what science are doing, so they will support science. So they will allow science, they will support funding for science and so on. That's part of it. The other thing is that you can really get a lot from the people. They can contribute to science, especially now with AI, people can, instead of just, I don't know what percentage of the world working on science, increase the percentage of people contributing to science. Of course, it's almost random who gets a good idea. So the more people are involved, the more we gain.
I love how you're framing it, that it's not dependent on your education. It's not dependent on where you're located. It's just dependent on you. And if you have a good idea, it doesn't matter where it comes from. And that's, I feel like that's the kind of gatekeeping that should be really just broken down. And I love that you're a champion of it.
If you, two examples, one of them, of course, kids that's the best, have the best ideas. This is, everyone knows that, right? And they're more speeded. Their brain is, like a sponge, and they're more creative in many ways. That's, everyone knows it, but we're not using kids to give us ideas. If brought to the right level, maybe they can do it. I'm sure they can. My daughter has amazing ideas, really crazy ideas. One of them we're even testing in the lab. It's really cool. And she had this idea when she was 6 or something. It's really, it's an amazing idea. And the other example, which is if you think about, sometimes people get Nobel Prizes for coming up with these amazing discoveries, and and they are so special. But that 10 other people were working on it at the same time, and in the end they got it, but maybe they were ahead of another group by 1 week, right? so many examples. Who is a scientist who was really ahead of his time, and we have is Mendel, Gregor Mendel, right? His paper, no one knew about it for 35 years, changed genetics completely. This is one person who we know was ahead of his time by 35 years and changed how we think about, and probably made the biggest discovery in biology. And he was totally isolated in a monastery in Brunn, He wasn't in a university. So he tried to engage with the university, but he always failed. Tried to be a teacher, he failed. So he was an outsider who made the biggest discovery and was really ahead of his time. So we have proof that this is how it works.
And now imagine with like the way the world is global with the internet, you don't have to be isolated. You could be exposed to these concepts and come up with the next world-changing idea.
I'm sure that this is what would happen.
I love that your daughter's ideas are being tested, by the way. I think that's incredible.
Yeah, I can even tell you her idea is really good. We're testing it. So maybe we'll end with that. I think it's a great idea. And this is an idea from really, now she is 12, but this is what she was 6 or something, 7.
That's awesome.
So the idea is, so the idea was, she said, you know how blind people have better hearing, for example, right? There's sensory compensation. So she said, what if, and this was during COVID, so many people were losing the sense of smell and so on, sense of taste. So she said, what if, we just inhibit a sense for a while, temporarily, so other senses would become stronger, okay? And then we bring back this other sense. Would the improvement stay? And we can make like super, people super sensitive, hearing well, although they now again see. We blind them for a while and bring the eyesight. Would the effect stay? And we said, okay, let's study this in C. elegans. There was a paper in C. elegans about sensory compensation, current biology by Ty Rabinovich many years ago. That let's do exactly that. In worms that don't smell well, they feel touch better, or the other way around. Don't remember. So we said let's just inhibit smell or touch temporarily, see whether they improve, and then and then bring it back and see whether they improve and stay. So this is the thing. And we can also do these prospective studies on people who had COVID who lost their, let's say, we didn't do that yet, but lost their sense of smell and say, did it improve other sensory modalities later on when they regained the sensory capacity? So that's the idea.
That's the chance to a 6-year-old with a concept of sensory deprivation and compensation. Like, what an incredible idea! Like, yeah, your daughter's awesome. That's also like I really connected with the video too of you were saying she's like the best thing in your life. I have a daughter and a son now too, and yeah, they're best things. And I'll never figure out how they inherited stuff. Like their heredity puzzle of what makes them who. Like especially even after this conversation as well, of like all the factors that could go into play. It's like we'll never know what makes them them, but that's part of the beauty of that journey. So as we wrap up, Oded, again, first of all, huge thank you for your time, for your expertise, for your kindness, not just on stream today, but to us and our family of communicating with us and agreeing to come on the stream. Is there any big future work that you're really excited about that we should look out for, and any, I guess, parting message that you want to leave the community with?
First of all, thanks a lot for hosting me, and it was a real pleasure. Pleasure is all mine, really. And we work on many different things, but in due time, maybe we'll do another stream and we'll talk about that as well.
We would love to have you back. When they have a new paper coming out and you want to talk about, the community will be here. Like again, like I said, whenever you get a preprint, we always show off your preprint. We've watched your TED Talks. I don't know, chat, how many times we've watched your TED Talk on transgenerational inheritance. It is an absolute joy and a privilege to have you on the stream, and thank you very much for sharing your knowledge, your kindness, and just everything. Thank you very much, Oded.
Thank you. It was great. Thanks a lot.
Thank you. We'll see you soon. Have a wonderful rest of your evening.
Thank you. Bye-bye.
Bye.